This presentation is on color reactions of protein. So these are the various tests that are performed to identify the different amino acids that are present in the protein. The list is as follows. First is Biochemistry test. This is a general test for proteins. So the observation is formation of violet color, which indicates the presence of protein. So minimum of 2 peptide bond has to be present for this test to be positive. What's the principle behind this test? Cupric ions in alkaline medium react with peptide having protein having 2 peptides to form violet colored complex, and this is a general test for proteins. So next is xanthoprotein test. Here yellow color changes to deep yellow or orange color. Now this indicates the presence of aromatic amino acids, mainly phenylalanine, tyrosine and tryptophan, and the reacting group is benzene ring here. So the principle behind this test is, so the benzene ring that is present in phenylalanine, tyrosine and tryptophan undergoes nitration when it is heated with nitric acid, and these are yellow colored derivatives. So on addition of alkali, these yellow colored derivatives get ionized and result in the formation of deep yellow or orange colored compounds. Now this phenylalanine, tyrosine and tryptophan are both glucogenic and ketogenic amino acids. Next test is Millen's test. Now this test is mainly done to identify the presence of tyrosine, and the reacting group is hydroxyphenyl group, and the observation is formation of red color precipitate. Now the hydroxyphenyl group of tyrosine, when it is heated with mercuric sulfate and sodium nitrite, it forms a red colored mercuric phenolate. Now this tyrosine is a non-essential amino acid synthesized from phenylalanine. There are so many compounds derived from tyrosine, and these include the catecholamines, adrenaline, noradrenaline, which increase the heart rate and force of contraction, increase the blood pressure. This dopamine is also derived from tyrosine. Dopamine is a neurotransmitter in cerebral cortex and basal ganglia. Thyroid hormones are also derived from tyrosine. So these are the compounds derived from tyrosine. Next is Hopkins-Coles aldehyde test. Now here purple or violet color ring is formed, and this indicates the presence of tryptophan, and the reacting group is indole group. The principle behind this Hopkins-Coles aldehyde test is that this tryptophan gets oxidized when treated with sulfuric acid and mercuric sulfate, and this oxidized tryptophan in turn react with aldehyde to form violet colored complex or the ring. Now tryptophan is an essential amino acid, and many compounds are derived from tryptophan. Now these include serotonin, again which is a neurotransmitter, melatonin which is responsible for maintaining circadian rhythm or the biological rhythm, niacin water soluble vitamin and its coenzymes NADP and NAD are derived from tryptophan. Alanine non-essential amino acid is derived from tryptophan. Apart from that, tryptophan gets converted to indole and scatol compounds and indican, which are excreted through intestine. Next is Akaguchi test. This is mainly for arginine. Bright red colored is formed, and the reacting group is guanidino group. So here the guanidino group of arginine react with alpha naphthol and alkaline hypobromide to form a bright red colored complex. Arginine is a semi-essential amino acid. Next is sulfur test. Now we get to see this brown or black colored precipitate, and this indicates the presence of sulfur containing amino acid. There are 3 sulfur-containing amino acids, methionine, cysteine and cysteine, but only cysteine and cysteine will answer this sulfur test, but not methionine. This is because the sulfhydryl group, that is SH group, is present in thioether linkage in methionine, which is very difficult to break. So only cysteine and cysteine will answer sulfur test. Now what is the principle behind this test? Now the cysteine and cysteine containing proteins, when they are treated with strong alkali, the organic sulfur splits and forms sodium sulfide. Now this in turn react with lead acetate to form brown or black colored precipitate of lead sulfide. Next is smallish test. Violet ring is formed at the junction of the two liquids. Now this indicates the presence of carbohydrate which is attached to protein, mainly indicates the presence of glycoprotein. So the principle behind this test is carbohydrates which are part of glycoprotein when treated with concentrated sulfuric acid undergo dehydration to form furfural derivatives. These derivatives in turn condense with alpha naphthol to form violet colored complex. The importance of this glycoprotein is that they act as cell surface receptors for the entry of biomolecules and also pathogens like bacteria, virus, protozoa to enter into the cell. The last test is ninhydrin test. Here blue or purple color is formed. Now this indicates the presence of amino acids with free alpha amino group. So all the amino acids answer this test except proline and hydroxyproline. They do not have free amino group, but they have imino group. So they will not answer this test positive. Instead of giving the blue or purple color, they form yellow colored complex. So what's the principle behind this ninhydrin test? So ninhydrin reacts with the alpha amino group of the protein to form blue or purple colored complex. Ninhydrin is a powerful oxidizing agent, and this causes oxidative decarboxylation of L amino acids. L amino acids are naturally occurring amino acids, producing an aldehyde, and in this process ninhydrin gets reduced. Now this reduced ninhydrin reacts with the amino group to form bluish or purple colored complex. So all the amino acids will answer this test forming blue or purple color except proline and hydroxyproline form yellow color because they contain amino group instead of amino group. So these are the possible questions that will be asked with color reactions of proteins. Apart from that, so you should know there are 10 essential amino acids, so 2 semi-essential amino acid, arginine and histidine, and 8 non-essential amino acids. All these non-essential amino acids are synthesized from essential amino acid, which has to be supplemented or taken through diet. So this was all about color reaction proteins. So this presentation was done by Dr. Preeti, Professor of Biochemistry from JJ Medical College, Davangere. Here are the links to other practical biochemistry. Thank you.